Good morning and afternoon, everyone. My name is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. Welcome to today's webinar presented by ASCCT and the European Society for Toxicology in Vitro, or ESTIV. If you're not familiar with our organizations, both of our societies host annual or biannual meetings and pursue other activities to promote the computational and in vitro toxicology fields. And we have a particular focus on supporting early career scientists and offering training and education. I wanted to provide a couple of brief announcements today. First of all, we have a few upcoming webinars on February 25th and March 11th. And you can register for those webinars on the ASCCT website, ascctalks.org. Secondly, ESTIV, it's having its biannual meeting in June of 2020 in Barcelona. And their abstract submission deadline is coming up. So please be sure to go to their website and submit an abstract. ASCCT here in the United States is helping to sponsor a QSAR 2020 meeting, a workshop on QSARs and environmental and health science. And I would encourage you to find more about the meeting on qsar2020.org and consider submitting an abstract as well. January 24th, this Friday, is the deadline. First of all, just a brief bit of housekeeping. This webinar is recorded. ASCCT and ESTIV members will have access to the recording shortly after the event. And everyone will receive copies of the slides from the presentations today. Your lines are muted to cut down on background noise. After each presentation, there will be an opportunity to ask questions using the question module. So again, we're going to have two short presentations, and after each presentation, there will be questions. So get your questions into the question module so that we can ask them right after the first and then second presentations. You could also raise your hand if you like. Either one will be fine. So today we have a special treat. We have two presentations given by exceptional early career scientists and ESTIV award winners. And I'm going to let ESTIV President Matthew Vinken say a few words here.
Thank you, Christy. So good morning or good afternoon to you, also from my side. So as you already heard, my name is Matthew Vinken. I'm the president of ESTIV, the European Society of Toxicology in Vitro. So if you were to be interested, which I hope, of course, you can go to our website, and then you will notice that we are the leading organization or society in Europe trying to promote in vitro and in silico toxicology at different levels of scientifically, educationally, and so on. Now, one of our priorities within ESTIV, and the same holds true for ASCCT, is to involve as much as possible the young people, the young scientists. Now, we try to do this in many ways, among which to grant a number of travel grants, but also poster awards or best oral presentation awards during relevant conferences. And actually, this brings me to the two speakers of today. So both of these ladies, they received an award during the previous Eurotox conference that took place in Helsinki in Finland in September 2019. So there was a Best Poster Presentation Award that was given to Lenny Kamelia for her work entitled A Battery of Animal-Free in Vitro Assays for Evaluating Prenatal Developmental Toxicity, Potency of High Complex Petroleum Substances. And then the second award was actually a travel grant that was granted to Laura Escriva. And both of them will present today their work. So with this, I would like to give the word back to Christy.
Great, thank you, Matthew. Without further ado, I'll go ahead and introduce our first presenter. That's going to be Dr. Laura Escriva. She's assistant professor at the Department of Preventive Medicine at the University of Valencia, Spain. She did her PhD in food science and toxicology and a postdoc at Karolinska Institute in Sweden thanks to the Europhora program from the European Food Safety Authority. Most of her research has been focused on mycotoxins, including in vitro, in vivo, and transcriptomic assays. And during her postdoc, she worked on risk assessment and endocrine disruptor identification. So I will turn the floor over to Lara now. Okay, Lara.
Hello?
Yeah, we can see your slides and I can hear you, so please go ahead.
Hello, good morning. Thank you for the nice introduction. So in this webinar, I would like to talk about the role of in vitro data for ED assessment. And to do that, I will start with a brief introduction on endocrine disruptors and the new regulation recently implemented in pesticides. And most of the presentation will be focused on the ECHA-EFSA guidance for ED identifications. And I will talk about mode of action analysis. And finally, I will present a case study on bisphenol AF that was the reason why I went deeper into the ECHA-EFSA guidance. And I will show you some preliminary results. So endocrine disruptors are exogenous compounds that interfere with natural hormone systems at very low concentrations, and they have been associated with several effects in humans and wildlife. And scientific understanding on the health impacts associated with endocrine disruptor has been growing in recent years. And in 2002, it was published a document on the scientific knowledge of endocrine disruptors, and then an update 10 years later. And it was concluded that EDs represent a special and challenging form of toxicity, being especially critical in developmental exposure. And close to 800 chemicals are known or suspected to induce endocrine disruptor effects. However, to demonstrate that a substance is an endocrine disruptor is not easy. The definition we use nowadays for endocrine disruptors was already published in 2002. So as you can see here, an endocrine disruptor is an exogenous substance or mixture that alters the functions on the endocrine system and consequently causes adverse health effects. And in 2013, the 3 elements required to identify an ED were described by the European Commission. So an endocrine disruptor substance has to show endocrine adverse effects, endocrine activity through an endocrine mode of action, and a biological link between the adverse effects and the endocrine mode of action. So the 3 conditions must be present to consider a substance an ED. In 2018, scientific criteria to identify endocrine disruptor substances were implemented in pesticides regulations, and at the same time, a guidance document for the implementation of these ED criteria was published by the European Chemicals Agency and the European Food Safety Authority. And this guidance is basically a protocol to identify if a substance has endocrine disruptor properties. So before going to the steps described in the guidance, it's important to mention that the ED criteria cover all the endocrine disrupting effects, but the guidance mainly addresses its modalities. This is estrogenic, androgenic, thyroid, and steroidogenic. And this is because there is better mechanistic understanding. There are some in vitro and in vivo standardized test guidelines, and there is a broad scientific agreement on the interpretation of the effects. So here you can see the flow chart for the assessment strategy presented in the guidance. And as you can see here, it consists of 5 steps. First, gather all relevant information, then assess the evidence, which includes evaluate relevance and reliability of the data, extracting and reporting the information, assembling and assessing lines of evidence for endocrine activity and adversity. And then the third step, initial analysis of the evidence, then mode of action analysis, and finally, an overall conclusion on the ED criteria. So briefly, step by step, for gather all relevant information, it should be performed by systematic review methodology. So basically, you have to find all the information about your model substance and select the relevant data by title screening and full text examination to build your dossier, which contain all the studies where you have to extract the relevant data. And then to assess the evidence, you have to evaluate relevance and reliability of all the studies in the dossier. And then all the parameters from each study should be reported using the Excel template provided in the guidance, which includes several columns, as you can see here, to capture all relevant information about the study design and the effects observed, for example, animal species or in vitro model, tested doses, etc. And each row reports the changes observed in a specific parameter within a specific study. And this means that you may have several rows for the same study. And also, it's important to report both positive and negative results. And once you have entered in the Excel all the data, there is some data metrics function that automatically rearranges the data to facilitate analyzing the information. And this is how the data matrix looks like from an example from the guidance. And as you can see, there are different colors based on the type of parameter or the type of data. And then these parameters are grouped into lines of evidence for endocrine activity and adversity. And lines of evidence for endocrine activity include information of in vitro mechanistic data and also in vivo mechanistic information. And when assembling lines of evidence for adversity, it should be considered all the effects related to its mediated parameters and parameters sensitive but not diagnostic of its. And the guidance provides some tables collecting the endpoints for all these sections. As you can see here, in vitro mechanistic in green, in vivo mechanistic in orange, and then its mediated parameters in blue and parameters sensitive but not diagnostic of its in purple. And this is an example on how looks like assembling and integrating the lines of evidence from the guidance. And then we move on to the third step, the initial analysis of the evidence, which consists on assessing if its mediated adversity or its endocrine activity has been sufficiently investigated based on the recommended assays. And the guidance describes six scenarios. Two of them lead to stop the assessment if its mediated adversity or endocrine activity has not been observed. One scenario leads to generating further data, and then 3 of the 6 scenarios leads to continue with the next step, which is the mode of action analysis. And basically, in these 3 scenarios, when potentially endocrine adversity or endocrine activity are identified, it's required to perform the mode of action analysis to establish the biological plausibility of the link between the its-mediated adversity and the endocrine activity, which, if you remember, is the third condition that has to show an endocrine disruptor substance. So then mode of action analysis consists of a series of biological events known as key events that results in a specific adverse effect. Key events are empirically observable and measurable events that are considered essential to induce the adverse effect. And as you can see in this figure, they are organized at different biological levels. The mode of action of an endocrine modality will normally contain some early key events which provide mechanistic data at molecular and cellular level, and then intermediate key events, and finally later key events at organ or system level, including the adverse effects. And finally, from the mode of action analysis, it's possible to draw an overall conclusion of the assessment, which is the last step of the process. So now that we have a general idea of the ECHA-EFSA guidance, we can start thinking on what's the role of in vitro data for ED assessment. And the first thing we have to think about is that there are only a few in vitro standardized assays for ED assessment available as OECD guidelines. You can see here some of them: estrogen receptor binding affinity, estrogen and androgen receptor transactivation, and steridogenesis assay. However, information from non-OECD studies should also be considered after evaluating the relevance and the metabolic capacity of the cell system, among other factors to be considered in vitro assays. And all these factors are collected in the guidance document on good in vitro method practices. But why are so important these in vitro studies? So as you have seen in the endocrine disruptor assessment, we have to collect and analyze data for adversity, but also for endocrine activity. And the information of endocrine activity should be collected from in vitro mechanistic data, which provide information on the mechanism through a substance could potentially cause adversity. For example, receptor binding or activation, inhibition of enzyme activity and cellular proliferation, and so on. But also in vivo mechanistic data, for example, changes in hormone levels in mammalian assays. And this mechanistic information is very important to understand what's happening at molecular and cellular level by setting the early key events that will lead to the adverse effects. And in fact, when we perform the mode of action analysis in the adverse outcome pathways, AOP context, it is recommended to include at least one key event at each major biological level. Therefore, in vitro data is required to feel and understand these early key events we were talking about. And moreover, from the regulatory perspective, the ED assessment is special because it is the only type of regulatory assessment, apart from carcinogenicity, where there is legal requirement to evaluate the mechanism, while for other effects, you only assess the adversity and the mechanisms are not investigated, or at least not required. Therefore, in vitro data is essential in ED assessment. And then in the last minutes, I would like to explain a case study that I had the opportunity to work thanks to the EU4A program from EFSA. And the work was conducted at Karolinska Institute. And here you can see the technical report, which is already available at the EFSA journal. But we are still working on the final data and also the manuscript, which I hope to be published soon. But I would like to show some preliminary results, mainly focus on the collected in vitro data. So the case study was based on the application of the ECHA-EFSA guidance for ED's identification to bisphenol AF as model substance. And it's important to mention that this ECHA-EFSA guidance was developed based on the ED criteria for pesticides, and its application was not intended for the assessment of rich chemicals. But the aim of the project was to explore the guidance application step by step on a non-pesticide substance and to identify the strengths and specific challenge in the process. Bisphenol AF was selected as model substance due to the amount of available data, the concern for human exposure and risk, and its similarities to bisphenol A, a known endocrine disruptor. The methodology, as you can see here, was basically to follow the different steps described in the guidance, and then we use additional tools for some of the steps. For example, we use Ryan tool for screening and SIRAP tool for reliability and relevance evaluation. So for gather all relevant information, the first step, we use 3 databases, Web of Science, PubMed, and Embase, by a single concept search, which means to capture all the information about the substance in one search by using the chemical name and its synonyms, for example, the CAS number and EPAC name. And we got 511 items after duplicates removal that were screened by title and abstract, obtaining 124 studies. And then in the full text examination, we finally arrived to 86 studies, which built our dossier. And then relevance and reliability of the data was evaluated in those 86 studies using SIRAP tool, as I mentioned before. And as it is shown in the slide, the great majority were reliable or partially reliable. And then in the table below, you can see the reliability results by the ID number of the, by the study, divided by the different type of studies. And it's easy to see here that more than a half of the studies were in vitro studies. In fact, we got exactly 53 studies, in vitro studies, from the 86 from the dossier. And these studies were performed in several cell lines from mouse, rat, monkey, and mainly human, providing in vitro mechanistic data by different assays. For example, estrogen receptor transactivation, estrogen, androgen, and thyroid receptor binding affinity, or steridogenesis in vitro. And it should be mentioned that only one study of these 53 were based on an OECD guideline. And then this figure shows an overview of the extracted parameters from the 86 studies from the dossier. As a reminder, each parameter represents a single row in the Excel file, and we got 510 parameters in total, divided in different groups. And then after rearranging all the parameters in lines of evidence, we arrived to 130 lines of evidence. And if we pay attention only on in vitro results or parameters, we got 171 parameters grouped into 20 lines of evidence. And here are some of the lines of evidence. For example, thyroid, androgen, or estrogen receptor activation, nuclear receptor activation, in vitro hormone levels, or gene expression and protein expression of hormone-related genes. And this information was very useful for the next steps of the procedure, specifically for mode of action postulation. But as I said, we are still working on the last steps of the assessment to finally arrive to a conclusion on bisphenol AF. So as final remarks, I would like to highlight that the ECHA guidance application on a rich chemical proved its practicality. However, some specific limitations were identified. For example, the guidance does not clearly define how to evaluate non-standardized studies. The standardized information requirements for pesticides include a number of standardized studies that are useful for DD assessment. However, this information could not be sufficient to complete DD assessment. And testing requirements under REACH are less thorough than for pesticides, adding extra limitations in the ED assessment of REACH chemicals. And the lack of information requirements is specifically critical in the case of in vitro and in vivo mechanistic data, since requirements are mainly focused on adversity. And although the adversity must be deeply evaluated, mechanistic data is completely necessary for ED assessment to evaluate the endocrine activity and to establish the biological plausibility of the link between the adversity and the endocrine activity. Finally, I would like to thank Steve and ASCCT for the award and also for giving me the opportunity to give this webinar. And for a program from EFSA and Karolinska Institute and all the colleagues that I work with that made possible all this work. And thank you very much for your attention. I will be glad to answer all the questions you may have.
Well, thank you so much, Laura. That was a really informative, nice presentation. Again, everyone, please type a question in the question box if you have one, or raise your hand. I can ask a question first. I was wondering what you think should be done in order to increase the use of in vitro assays or the conduct of in vitro assays for endocrine assessment in a regulatory context. It looks like your case study, there were quite a few in vitro assays conducted, which is good to see. But I wonder if that information is being effectively taken into account in a regulatory context.
I think they are every time more and more considered, but I think it's important to, of course, standardize these studies, not only by guidelines, but at least to make sure that they are reliable and they are comparable between the different. So it's good to standardize procedures. And also in the context of ED assessment, I think it's already being done to standardize more studies specifically for endocrine disrupting endpoints, because now there are only very few, and there are, for example, none for thyroid modalities. So it's important that we got more and more every time.
Yeah, great. Okay, one question coming in. Fantastic presentation, Laura. Could you clarify how study quality is being used, specifically for studies that are not reliable? Are they still being considered in developing conclusions?
Yeah, study quality, I mean methodological quality and reliability. So in this case, since the studies also were not OECD guidelines or standardized, we use the SIRAP tool, which is a tool, online free web tool that with some criteria, but not based on if the study is standardized or not, based on the methodological procedure. And we use SIRAP for the evaluation of all the studies. And also it's interesting because there is SIRAP for in vitro and also for in vivo. So we can evaluate with different specific parameters. In the presentation, there is a link if you want more information about the tool.
Okay. Just let's see real quick. So follow up on that is whether with the SIRAP tool, have you compared what the reliability and relevance that the tool gives you and what a person or an expert assessment of relevance and reliability would say? Has there been a comparison of those? Do you know?
Do you mean if we have compared the results with the
so the tool gives you an assessment of the reliability and the relevance of the studies, and you could also get that with a person, looking at all of the studies and determining the reliability and relevance. Is there likely to be a difference between those two approaches?
Yes, I mean, it depends, because the good point is that with the SIRAP, you have some standardized questions, and it tries to make it more objective. So yeah, I think it's easier if you have some guidelines and some specific criteria that you only have to mark if it's fulfilled or not fulfilled or partially fulfilled, than only by some expert judgment just evaluating the study.
Mm-hmm. Great. We have two people who want to know whether you have any opinion on the use of in silico models for assessing endocrine disruption.
I think they could be useful as and also supporting the evidence. But as I mentioned, finally, it's important to have information at different biological levels, and of course, in silico is it could be useful for support the results we have by in vitro and different biological levels. But it's true that it's also a bit difficult to know how to consider this data when you are, for example, doing this assessment. It's not easy, and the same for epidemiological data. I mean, of course, you have to consider all the information you have, but then it's sometimes a little bit tricky to know how to consider this data. But the thing is that as more as biological levels of organization you have, the information, it's more, the evidence will be higher.
Great. So we have a few more questions, but what I think I'm going to propose to do is, just so that we make sure we have enough time for Dr. Kamelia's presentation, is maybe we'll move to her presentation and do some Q&A, and then if we have extra, we can come back to some of these questions. So thanks everyone for all your questions. I'm going to turn now to Dr. Lenny Kamelia, and can't do two things at once, so I'm going to give her the presentation rights and then introduce her. So Dr. Kamelia just recently obtained her PhD in toxicology and is currently working as a postdoctoral researcher in the Division of Toxicology at Wageningen University and Research in the Netherlands. Her research interests include in vitro and alternative methods, reproductive and developmental toxicology, PBPK modeling, and risk assessment. And so please go ahead.
Hello, thank you, Christy, for the introduction. And I also would like to thank Steve and also ACCT for the opportunity today, so I can share a part of my PhD work for this webinar. And also, of course, I would like to greet all of you who made time to join this webinar. And here is the content of today's presentation. I will start with giving background information of my study, will be followed by the research approach applied, and then I will walk you through some results and discussion points, and then finally the conclusions. So let's get started. As you all know, rich legislation, they require substances that are produced in or put onto the EU market at a volume above 100 tons per year to be tested for effects on prenatal development. And this also applies to highly complex petroleum substances. If this testing is done following the current OECD guidelines, which is OECD 414, then consequently, a huge number of experimental animal and also resources are needed. So from that point of view, the application of alternative testing strategy that can cover the relevant mode of action of test compound of interest, of course, is of high relevance and important. Speaking about petroleum substances, so what are they and what they are consist of actually? So petroleum substances are produced by either atmospheric or vacuum distillation of crude oils, as illustrated here on the left part of the slide. This crude oil distillation will generate different types of petroleum substances products. And of course, they are produced with different usage purposes. And by definition, and also under REACH, petroleum substances are characterized as UVCBs. That stands for unknown, variable composition, complex reaction products, and biological materials. They contain at least thousands to millions of molecules, and that includes polycyclic aromatic hydrocarbons, or PAH in short. Then why it is very interesting to research about this PAH, especially in relation to developmental toxicity of these complex substances? This is because a available in vivo study showed that developmental toxicity, as observed with some petroleum substances, is associated with the presence of specific group of PAH in these substances. Illustrated here, for example, heavy fuel oil, or HFO, that can contain up to 30% PAH, mainly 3 to 7 ring PAH, they are tested positive for developmental toxicity in animal studies. On the other hand, when similar studies were conducted by using either highly refined base oil or GTL products, and both of these products contain no aromatics and no PAH, they are tested negative for developmental toxicity. So it can be hypothesized that there is a causal relationship between developmental toxicity, as observed with some petroleum substances, and PAH present in these products. To test this hypothesis, we selected and applied a battery of alternative in vitro assays that include the embryonic stem cell test, or EST, zebrafish embryo toxicity test, or Z, and aryl hydrocarbon receptor assays. So the idea of combining these assays is to evaluate the developmental toxicity potency of a series of petroleum substances, and if possible, we compare that with the potency observed in vivo. To that purpose, we selected a series of DMSO extracts of petroleum substances within and across product category. So as illustrated here in this bar graph, you can see that the selected samples contain a variation for the type and level of PAH they contain. In addition to the PAH containing petroleum substances extracts, we also tested samples that are known to contain no PAH, like highly refined base oil and GTL products. So basically, these samples serve as the control negative substances in our study. So now I'm going to walk you through the results obtained in each in vitro assays that I applied. The first one is the EST. So in this EST, I use mouse embryonic stem cell line D3, or you can call it ESD3 cells. And this cell can spontaneously differentiate into contracting cardiomyocytes. So the ability of test substances to inhibit this spontaneous differentiation is then used as the endpoint of the assays. So here I would start the assays. The assay itself, it lasts for 10 days, and it starts with the hanging drop culture. So basically, I hang the cell suspension upside down, so to create what's so-called embryoid body or spheroids here. So the formed spheroids will grow in size, and the exposure medium is refreshed 2 times during this period of 10 days. On day 5, the spheroids will be transferred into the 24 well plates, so 1 spheroids per well. And on day 10, the number of well containing contracting cardiomyocytes is then counted. So here I give an example. If a well contain beating cells or beating cardiomyocytes, then score 1 is given. Then if not, score 0 is given. So here are the obtained results. So if you combine all the results obtained from this ESD3 cell differentiation assays of the EST, this is the result how it looks like. So in short, you can see that all PAH containing petroleum substances extracts show inhibition of this stem cell differentiation in a concentration dependent manner. And most importantly, this occur at non-cytotoxic concentration. On the other hand, all PAH-free extracts like gas to liquid products and highly refined base oil extracts, they are tested negative in the EST. Furthermore, we also plot the potency obtained in the EST against the pH content. So we can see the correlation between pH content and in vitro potencies obtained in the EST. And here you can see again that in vitro potency in the EST is proportional to the level of 3 to 7 rings pH present in this sample. The second assay applied is the Z. In this assay, fertilized zebrafish embryo are carefully selected and then exposed to different concentration of test substances. 124 well plate, I use it to test 1 concentration, like in the EST, and also 1 embryo per well. And every plate also has their own internal plate control that actually used for the validity of the test. Following exposure, the development of exposed zebrafish embryo are scored using what's so-called general morphology scoring system, or GMS. The GMS includes scoring on development of eyes, tail, pigmentation, heart and circulation, the development of fin, and also mouth. So the scoring was done every 24 hours, starting from 24 HPF or hours post-fertilization to 96 HPF. In addition to this GMS, some malformation-related endpoints were also scored. And these include, for example, edema formation, deformation of tail and body, and also the presence of swimming bladder. Here's an example how the developing zebrafish embryo looks like over the exposure period. And here I compare the negative control with one of the test sample, the HFO extracts, sorry. At 24 HPF, here you can see that, I'm not sure if the video works, I hope so. So at 24 HPF, for example, you can see that the exposed zebrafish embryo has a less frequent movement compared to the negative control. And at the 48 and 72 HPF, you see that there was a pericardial edema formation. And the hatching also was obstructed at the 72 HPF. Some of them survived, but some of them also died at 96 HPF. So again, if you then plot all of these results from SAT together, you can see that all of PAH containing petroleum substance extracts induce again concentration response curve of growth retardation in the zebrafish embryo development. And it's good to mention that only results obtained from the 96 HPF are plotted here in this graph. And again, when you want to then try to make a correlation between potency obtained in the Z and the pH content, you can do that by simply plotting them together. And here you see the potency is correlated or proportional to the level of 3 to 5 ring pH. And one of the difference between the result or this correlation between the EST and Z, you see that with the EST, it correlates best to 3 to 7 ring, and with the Z, it correlates best to 3 to 5 rings. And one of the explanation for that is that in the EST, we use serum-based medium. In the Z, we use saltwater-based medium. Then, of course, this may affect the bioavailability of the test compounds during the exposure period. And then, of course, when you have done in vitro study, you always would like to compare the result to in vivo data when it's available, to see actually how good is the prediction or correlation between the obtained in vitro results to in vivo data. So although here the available in vivo data is a bit limited, but as shown in these two graphs, you can see that there's a good correlation between the obtained in vitro potencies in both EST and Z with available in vivo data. So I have mentioned to you first that there's a good correlation between the pH content present in each petroleum substances extract that I've tested with in vitro potency in both EST and Z. And on top of that, there's also a good correlation between in vitro potency obtained and in vivo potency observed. So then, of course, it is tempting not to use such correlation to then try to predict the developmental toxicity potency of untested petroleum substances extract. Of course, that belong to the same product category that I have tested. Here is the example how the proof of concept, how it can be applied. In the blue column is the value of experimental in vivo potency of sample 175. And in the orange column is the predicted value using the previously mentioned correlation models, or we can call it like structure activity relationship models. So here is clearly seen that you can clearly see that the experimental in vivo potency is highly comparable to the predicted one that actually differ only 1.1-fold. In addition to the EST and Z, we also would like to study the mode of action underlying the developmental toxicity of the PAH containing petroleum substance extracts. So for that, we applied a panel of Calyx assays, including the HR Calyx assays. And here you can see that all PAH containing petroleum substances extracts show HR-mediated activity. And most importantly, this HR-mediated activity correlates well to the result obtained in the Z and EST, suggesting actually the role of this specific receptor in mediating this effect. So to prove that further experiment was conducted by co-exposing the cells with petroleum substances extracts in the presence of an AHR antagonist, and we chose trimetoxyflavone. This was done in both the EST and Calyx assays. And here are the results. On the left, you see the results from EST3 cell differentiation assays, and on the right, you see the result from AHR Calyx assays. So in short, co-exposure of cells, either in EST or AHR Calyx assay, neutralize first the petroleum substances extract induced developmental toxicity, and the second, HR-mediated activity as tested in HR Calyx. And for the pH-free extracts like GTL and highly refined base oil, they are tested negative with or without the presence of HR antagonists. Therefore, it can be concluded from this experiment that HR activation at least is one of underlying mode of action for the petroleum substances extracts induced developmental toxicity. And here you can also use a PCA biplot to represent and illustrate the data obtained that include the pH content and also potency obtained in the Z, EST, and also AHR calyx. So I would like to share two main messages here. The first one is that you can clearly see that petroleum substances extracts that belong to the same category, they are all clustered in the same quadrant of the PCA biplot. And of course, it can be clearly seen that the types and amount of the pH present in each sample actually determine their in vitro potency. To conclude, the developmental toxicity of the highly complex petroleum substances can be assessed using the presented alternative testing strategy. And this potency is proportional to the amount of specific group pH present in these complex substances. And regarding the mode of action, at least the HR activation actually play an important role in mediating this effect. And also a good correlation also exists actually between in vitro result and in vivo data. So then people always also ask the question, what will be the implication of this result? How can you use such a result first to support the risk legislation, for example, and perhaps also in relation to the 3R principle in toxicity testing? Of course, the obtained results can further be used to assist the grouping of these petroleum substances based on their analytical and bioactivity profiling. And this will also facilitate read across and the selection of the worst case representative per product category, where, for example, in vivo testing is needed as a last resort. Last but not least, such alternative testing strategy will definitely contribute to the use of actually the 3R principles. So then we can reduce the required animals to evaluate the developmental toxicity potency of petroleum substances as required by REACH. I dedicate my last slide of my presentation today to acknowledge both Concave and Wageningen University for funding my PhD project. And also thanks to all the people who are actively involved in my PhD project. It was a great pleasure to work with them. And also I would like to thank Steve for the award and to all the audience that's spending your time listening to my presentation. And with this, I'm happy to take any questions you may have.
Wonderful. Thank you, Dr. Kamelia, for this presentation. It's very nice. While people are adding their questions, let me ask, you answered my question that I typically ask, which was about about uses. So what are you working on now? Are you working on extending this work, or do you have plans to do that?
Yes, so basically now we are, now we have also some new PhD helping, so now I'm a postdoc. So we would like to extend this research. For example, always the question comes from reviewer or also regulators that because I use the MSO extracts, for example, testing these substances, because that's for a reason. There's also study have done in the past for example, MS tests. When you then compare using net material compared to the DMSO extracts, actually the use of DMSO extracts increase the interaction between the in vitro assays applied with these materials. Mm-hmm. So therefore, it's kind of helping to dose them in vitro. But also in addition to that, of course, then actually we are not limiting our research only on PAH, because we have to also acknowledge PAH also belong to polycyclic aromatic compounds as a whole. So there's also a possibility that there are other type of constituent, for example, the substituted one, like alkylated one, that could also contribute to this type of toxicity. Mm-hmm. So therefore, this PhD and also together with me, we are going to use the same battery, also to look at the possibility to add maybe other in vitro assays to be added to this battery. So then we can further prove by using more individual compounds. So then instead of complex substances, so then to prove this hypothesis further. So that will be the plan. And also try to use such PK modeling but only for individual compounds, not the complex substances, to see how good is the actually in vitro study that we have conducted in relation to if you then want to extrapolate that to human situation, then you need to take into account kinetics as well. So we try to also include that for the future studies.
Okay, great.
Will that answer your question?
Yeah,
I hope
yeah,
so.
definitely.
Yeah.
We have a couple questions coming in. Again, just to advise everyone, go ahead and type your questions into the question module. One question is, how can you confirm that the proportion of a UVCB not extracted into the DMSO has no developmental activity?
Yes, so I already mentioned shortly, I think, about the use of DMSO extracts. So what actually left after the DMSO extraction, we call it a raffinate part. And that mainly contain highly alkylated PAH, or actually very more polar part of the, and we know that from both also conducting by doing in vitro, and also there are some in vivo studies show that if actually it's just mainly highly alkylated PAH, so they are not bioavailable. And also that if it's mainly naked, like 1 to 2 rings benzene rings, so then actually they are also not really related to developmental toxicity. So therefore, again, maybe in my PhD research, I focus more on this 3 to 7 ring PAH. So perhaps this has to be extended to 3 to 7 ring PAC, so the polycyclic aromatic compounds as a whole. So then to clarify this further. So I'm not saying that, but actually using, I can say so far that using the DMSO extracts, apparently we predict quite well the in vivo developmental toxicity. So both for increased resorptions endpoint and live phytosis per liter endpoints. So for that, we predict very well.
Okay, great. Another question is about whether a similar assessment of petroleum substance extracts have been conducted for estrogen receptor assays.
Yes, so basically, yeah, it's been published in
the 2 webinars.
Yes. But again, I think so I also published that data in Toxicological Sciences in 2018. So I use a panel of the Calux assays that include ER, so estrogen receptor, thyroid receptor, and then what else? Progesterone and androgen. So I have tested indeed this extract in this a panel or battery. But when you then try to analyze, of course, I mean, when you then look at endocrine activity, I think it should not be translated immediately into endocrine toxicity, right? So you have to always then correlate the data. So then apparently the data from the AHR correlates very best to what actually in vivo data related to developmental toxicity, and also the data obtained in EST and Z. So therefore, I only present here, of course, due to the time, the data from AHR, but the dose sample indeed has been tested also in the panel of this receptor-mediated that include estrogen, androgen, progesterone, and thyroid. And I think I can refer to my publication in 2018 to see, I present that as a heat map, I think, for such activity, but indeed only the HR-mediated activity that very prominent in relation to this developmental toxicity endpoint.
Okay, great. And maybe what I can do is share the reference with everybody.
Yeah.
Let's see, one more question about whether, so they're thinking about animal studies versus in vitro studies, and wondering whether you think that animal studies are more reliable for developmental toxicity of these materials.
Well, I think it's a very tricky and difficult questions, because then from my perspective of when I did all of this battery, with available in vivo data that we have, apparently this battery first can be used for sure for screening, for example, and prioritization purposes, and also for selecting worst case sample per product category. But I'm not saying that, of course, then which one is actually the gold or better. But I think so far the data that we have, at least the use of this battery, so you have to do it in a battery, not like as a standalone assays, at least it supports the in vivo findings. So with that, therefore, I propose that if you want to test the same extracts that belong to the same category that I have tested so far in this project, then I'm quite confident to say that it predicts quite well. So then again, come back to the questions about do I think actually the in vivo study then still serve best? In this case, I may be confident enough to say not always.
Sure, absolutely.
Yeah.
Okay, here's a question about the embroid bodies. Wondering if they could differentiate into other cell types, because right now you're looking at cardiomyocytes, and so they're curious about other organ structures.
Yes, so these stem cells, so mouse embryonic stem cells, they can also be differentiated into neurons, for example, and other type of cells. So in theory, of course, it's possible. I know that there's one group in other university, they use it mainly for neurodevelopmental toxicity. So it's possible. So in our case, we start with beating cardiomyocytes because then it's the most, how to say, easier compared to the other differentiation process. Because for differentiating them into neurons, it takes, I think, almost a month or 3 weeks. So it's possible. So you just need to add different supplements to induce such a differentiation, but it's possible to do that for the other type of cells.
Okay, right.
Yeah.
So we have a few more minutes, and I want to try to get to someone who has their hand raised. So they've been very patient, and I'm not sure, to be honest, whether it's a question for Laura or Lenny. So I'm gonna just, Mr. Monaz Maisoruela, I'm going to unmute you. If you would like to answer your question, you have your hand raised. Please go ahead. Monaz Maisoruela. Okay, maybe they could type their question because I'm not hearing them. Okay, we have another hand raised, Kumar Sambhav. I'm going to unmute you. Would you like to ask your question? Nope, hand went down. Okay, forgive me. We want to make sure they have an opportunity, but I think what I'm going to do, we have 3 or 4 questions, but we can't get to all of them, and for you both. So what I'm going to do is ask that maybe we can send them by email, and then we can maybe have some post-webinar exchange.
That's a great idea. I think I like that idea.
Okay, thank you so much for being willing to do that. So I'm going to close out the webinar now. I want to thank all of you for attending and for your attention, and I want to thank our presenters, both of you, for the time to put together the presentation and to give it and to respond to all these wonderful questions. I think it was a really productive hour, and thanks again, and I hope you all have a great afternoon. Bye, everyone.
Bye.
Hi.